Welcome back to The Deep Dive, the show that takes a concept you think you know and reveals, well, a whole universe of surprising insights beneath it. Today we're plunging into a word that's probably on your grocery list, maybe in your fridge, or perhaps even lurking in the back of your mind after, a delicious meal. Yeast. We hear it, we use it, it's almost an everyday term. Okay, let's unpack this. But how much do you really know about what yeasts are, where they come from, and just how incredibly diverse they truly are? Prepare to have your understanding reshaped by the surprising complexity behind that simple, familiar name. I know mine certainly was when I started digging into our source material for this deep dive.
Indeed, and our mission today is really to move beyond that superficial understanding. We'll be guiding you through Chapter 6 of Bryce Kendrick's essential work, The Fifth Kingdom. It's aptly titled Yeasts, Compact Polyphyletic Extremophile Fungi. We're going to pull back the curtain on the hidden world of yeasts and hopefully dispel some common misconceptions you might hold. What's fascinating here, as you touched on, is that the term yeast isn't really about a single family on the fungal family tree. Instead, it's more about a lifestyle or a form that fungi can take. That's the central paradox, isn't it? These organisms are invariably microscopic, tiny little things, yet their impact on our world is profoundly, almost unbelievably large, from baking our bread to brewing our beer and, yes, sometimes even causing serious diseases.
Yeah, so if you're like me, you probably picture yeast as this, like perfectly oval, single-celled organism happily bubbling away in dough or fermenting grapes, a simple, helpful little guy. But that, as we're about to discover, is a heavily simplified view. Our source material tells us that currently, scientists have identified about 1,500 species of yeasts.
1,500.
Yeah, distributed among about 100 different genera. That's a lot more than just the Saccharomyces cerevisiae in your bread. And while they're incredibly useful for bread and alcohol, they also have a darker side, as the chapter puts it. Some spoil food, which leads to significant economic losses, and others, like the infamous Candida albicans, cause potentially serious human diseases. And here's where it gets really interesting, something that completely challenged my own preconceptions. We're often taught that yeasts are just single cells that don't produce hyphae, those long thread-like structures you see in most other fungi.
Right, the filamentous growth.
Exactly. But Candida and quite a few other yeasts clearly do produce hyphae alongside what we typically think of as yeast cells.
That's the dimorphism.
Ah, okay.
Yeah.
And in fact, these yeast cells themselves are essentially a type of asexual spore, which mycologists call conidia. And they develop in what's described as blastic acropoidal branched chains. So try to visualize a chain of pearls, maybe.
Yeah, that's a good way to think about it.
But each new pearl sprouts from the tip of the previous one, creating this growing, branching string rather than just a simple row. It's a far cry from just those neat little ovals we imagine.
Absolutely. And that ability to switch forms, that dimorphism you mentioned, is precisely why understanding this diversity, both in their shape and, what they do, is so crucial, especially when we consider their economic and medical importance. For a long time, zymologists, the yeast experts, faced a significant challenge. Many yeasts look microscopically identical, but physiologically they're very different.
Right, like trying to tell apart identical twins just by looking, when their internal workings are totally different.
Exactly. So the initial taxonomic schemes, how they tried to classify them, had to rely on physiological tests. Things like their ability to ferment or assimilate various sugars, their specific nutrient requirements, what vitamins they need, what nitrogen sources, or even their resistance to certain antibiotics. But as our scientific understanding grew, well, so did our toolkit. More recently, really sophisticated techniques have been pressed into service to truly unravel their complexity. We're talking about things like magnetic resonance analysis of cell wall components, electrophoretic enzyme analysis, cytochrome spectrophotometric analysis.
Wow, that's quite a list.
It is. Serological tests, DNA reassociation, analyzing the DNA-based composition, and now entire yeast genomes have even been sequenced. So to answer the question of why we need all these advanced tools for something that seems so simple, well, it's because their apparent simplicity on the surface hides this immense biological complexity. It demands a truly cutting-edge scientific arsenal to accurately track and understand them. They are, in a way, microscopic masters of disguise.
That's a fantastic phrase for it. Microscopic masters of disguise. And it really makes sense when you consider how many different ways they reproduce and grow. It's not just a simple budding or fission, like you might learn in basic biology. As we just heard, the assimilative yeast cells are now understood as conidia, essentially, asexual reproductive spores or single-celled propagules. And this chapter outlines several distinct types of conidiogenesis, which is just a fancy term for the process of forming these conidia.
Uh-huh.
Trying to visualize these without a diagram is a fun challenge, so let's walk through a few, and I'll try to paint a clear picture for you. First, think about multilateral budding. This is what many of us probably imagine for Saccharomyces, right? The common baker's yeast.
The classic example.
Yeah. Picture a spherical cell, and from many different points on its surface, it pushes out small daughter cells, the conidia. Each site leaves a little scar, like a little mark. But importantly, only one daughter cell forms from each point. It's like a balloon, sprouting mini balloons all over its surface, one at a time from different spots.
A good visual.
But what if a yeast prefers a more focused approach? Let's turn our attention to apiculate or bipolar budding. Here, imagine a longer, more oval-shaped cell, maybe lemon-shaped, like Saccharomycotes. It doesn't bud from everywhere.
Right.
Instead, it buds repeatedly from both ends, both poles, extending its growth in a continuous fashion. It's almost like it's stretching itself out, budding off new cells only from those specific points.
And that focused growth strategy is a stark contrast to what we often, maybe incorrectly, call fission yeasts like Schizosaccharomyces. They also extend their growth, but on a much broader base. They effectively divide into two, like a symmetrical split.
Like pinching something in the middle.
Exactly, almost like pinching a balloon in the middle until it separates into two equal halves. It's a completely different mechanism from budding, but the end result is still, an increase in cell number. Then you have phallic arthric conidia, typically found in hyphal yeasts like Geotrichum. This one's quite different. Here, the fungal filament, the hypha, it simply breaks apart into individual, often sort of box-like conidia.
So fragmentation.
Yeah, imagine a long string of beads just fragmenting into separate pieces. There's no complex budding process. It's more about breaking up an existing structure. Another fascinating one is blastic symponial conidiogenesis. You see this in some Basidiomycetes yeasts, like Cryptococcus. Visualize a cell where conidia are formed one after another, but each new one pushes the previous one slightly aside.
Oh, creating a kind of zigzag.
Precisely, a zigzag pattern of growth. It's almost like a microscopic assembly line, but with a slight directional shift each time a new spore is made. And finally, let's touch on blastic phyletic growth. This is found in genera like Rhodotorula or Sporobolomyces. Here you have a specific, often flask-shaped structure called the phyla.
Yeah.
And it continuously pinches off new conidia from its open end, almost like a factory of production line. Each spore pops off, and another immediately starts forming from the same point.
Wow, okay, so that's a lot more going on than just a simple bud. And the chapter mentions that sometimes these asexual forms, what we call anamorphic forms, can switch into a sexual teleomorphic mode.
They can reproduce sexually, too.
But often this happens without a dikaryophase. Now, you mentioned that earlier, but for those of us who aren't steeped in mycology, what exactly is a dikaryophase, and why is its absence in most yeasts significant?
That's a really crucial distinction, actually. In many other fungi, the dikaryophase is a really important stage in sexual reproduction. It's where a cell briefly carries two separate, genetically distinct nuclei, one from each parent, essentially before they finally fuse together. Think of it as N plus N.
Okay, two separate nuclei hanging out together.
Right. So when yeasts switch to a sexual mode without a dikaryophase, it means their genetic fusion process is often much more direct and, well, simpler. The nuclei fuse almost immediately. This sets them apart from the typical reproductive cycles of other, more complex fungi, and it points towards a unique evolutionary path for many yeast groups. These morphological details and reproductive strategies are incredibly important because they offer profound clues to the underlying phylogenetic diversity, their evolutionary relationships. Some yeasts, for example, form endogenous meiospores. Those are the sexual spores inside structures called meiosporangia. Meiosporangia, got it. Now, these meiosporangia are karyologically, meaning genetically, in terms of chromosome behavior during meiosis, exactly comparable with asci, which are the spore sacs of Ascomycetes.
Okay, so they're like asci in function.
Functionally similar in producing spores after meiosis, yes. So if we connect this to the bigger picture, while these yeast meiosporangia typically contain 4 spores, just like many asci, their cell wall chemistry is distinct from mainline Ascomycetes. And critically, they are never produced within a larger fruit body or ascoma, unlike most other Ascomycetes. They're just formed singly or maybe in short chains. This again hints at a unique lineage.
And that ability of some yeasts to produce hyphae, as we mentioned earlier with Candida, that seems absolutely key to understanding their adaptability. So we're really seeing how that simple idea of yeast as just one thing is kind of crumbling, aren't we?
It really is.
And it's not just about Ascomyces either, is it? These yeast forms pop up elsewhere.
Exactly. The story gets even broader. Other yeasts are actually members of the Basidiomycota. That's a completely different major fungal phylum, the one that includes mushrooms and rusts and smuts. Some of these, like Sporobolomyces, produce exogenous spores, meaning formed externally borne asymmetrically on little pointed outgrowths called sterigmata. And these spores are forcibly discharged, shot off. The mechanism involved is clearly that of the basidium, the characteristic spore-producing structure of Basidiomycota.
Even if the spores themselves are asexual.
Even if these particular ones are asexual mitospores, yes. The ejection mechanism is the giveaway. Others, like Cryptococcus, produce those blastic phyletic or blastic sympodial conidia we described earlier. What's even more telling is that some hyphal Basidiomycetes yeasts actually make clamp connections.
Ah, clamp connections. I remember those little bypass loops in the hyphae.
Exactly, those little bridges. They're a classic diagnostic feature of many Basidiomycota, involved in maintaining the dikaryotic state during growth. Finding them in a yeast-like fungus is a strong indicator of its Basidiomycete affiliation. This really brings us squarely to the polyphyletic spectrum, which highlights that yeast isn't a single lineage, but a form that has evolved independently in many different fungal groups.
Right, convergent evolution.
Precisely. It's like different evolutionary paths have all converged on this successful, often single-celled lifestyle. Let's maybe trace some of these paths outlined in the chapter. We can even start with the Zygomycota. Think about Mucor rusi, for instance. This organism is fascinating because it can switch between being hyphal, filamentous, and having a yeast-like morphology simply based on environmental factors.
Like what kind of factors?
Things like changes in carbon dioxide levels or the type of nutrients available. High CO2 can make it grow as yeast cells. This is a great illustration of how morphology isn't always rigidly fixed by genetics. It can be a highly adaptive response to the environment.
Okay, so Zygomycetes can do it. What about the Ascomycota? That seems like the main group we associate with yeast.
It is a major group, yes. And within the Zygomycota, the yeast story becomes even more intricate. The yeast form pops up in several distinct branches here. You have the subphylum Saccharomycotina, which is often called the true yeasts. This class, Saccharomyces, includes many of the familiar genera like Saccharomyces, Saccharomycodes, also Dipodascus, which has Geotrichum anamorphs. Those are the asexual forms. We also find Saccharomycopsis with its Candida anamorphs and Hanseníaspora with Cloacra anamorphs. Lots of connections there.
And it's within this group, the Saccharomycotina, that we find Candida albicans, the one that famously forms aerial hyphae and causes candidiasis. So, expert, this brings us back to that question. If it forms hyphae, is it really a yeast? What's the definitive evidence that still groups these as yeasts, but also makes them distinct from the regular filamentous Ascomycetes?
That is the million-dollar question, isn't it? And the chapter provides clear answers based on a combination of features. The evidence groups these ascus-forming yeasts as distinct for several key reasons. First, they generally lack that dikaryophase we discussed.
Right, the N plus N stage.
Second, their ascus-like meiosporangia are produced singly or maybe in chains, but never within a larger complex structure called an ascma, which is typical for many other Ascomycetes. Third, their cell walls have a different chemical makeup, generally less chitin and more mannan compared to filamentous Ascomyces. And fourth, their septa, those internal cross walls in the hyphae when they form them, often contain many narrow micropores instead of the single, larger central pore seen in many other Ascomycetes.
Ah, okay, so it's a whole suite of characteristics.
Exactly. So yes, they are yeasts in form and lifestyle, but with this unique underlying biology. We also find other Ascomycota showing yeast phases, like Taphrina and the Taphrina which causes things like peach leaf curl. And histoplasma in the Pizziziomycotina, this one is really interesting. It's filamentous in culture, like in the lab, but becomes yeast-like when it's growing inside human tissue, causing histoplasmosis, another striking example of that environmental adaptation or dimorphism.
Okay, so Ascomycetes are complex. What about the Basidiomycota yeasts? You mentioned Cryptococcus and Sporobolomyces.
Right, let's transition fully to the Basidiomycota, a completely different phylum. In the subphylum Agaricomycotina, class Tremelomycetes, we encounter genera like Phylobasidium and Phylobasidiella. Their anamorphs, their asexual forms, are often Cryptococcus. What's fascinating here is their cell wall chemistry again. These yeasts have chitin-mannan walls, like the Saccharomycetes, but they also contain sugars like xylose or fucose, which are entirely absent in the Saccharomycetes. It's a key chemical
Like a
difference.
biochemical fingerprint.
Precisely. Cryptococcus neoformans, for example, is a major pathogen causing cryptococcosis. It forms those blastic sympodial or blastic phyolitic conidia we talked about. And importantly, when their teleomorphs, their sexual forms, are known, they produce clamp connections and basidium-like structures, classic Basidiomycota traits. You'll also find trichosporin in this group, an anamorphic yeast whose hyphae exhibit dollopore septa.
Dollopore septa, those barrel-shaped pores.
Yes, those unique complex pores in the walls between cells, characteristic of many Basidiomycota, allowing for controlled flow of cytoplasm and even organelles. Finding them is another strong clue. Then there's the subphylum Proteinomycetina, specifically the class Microbacteriomycetes. These are often called the red yeasts. Because they contain carotenoids, those are the vibrant pigments that also give carrots their color. So these yeasts often form characteristic pink or red colonies. Rhodotorula, for instance, is a common one, forming blastic phyolitic conidia. And Sporebolomyces cells develop those sterigmata, from which they forcibly eject asymmetrically born spores, the ballistospores. Again, that spore-shooting technique is very reminiscent of a basidium, but critically, these are asexual mitospores, not sexual ones in this case.
So the mechanism is similar, but the spore type is different.
Exactly. And finally, we can't forget the subphylum Ustelaginomycetina, which includes the smut fungi. Even these plant pathogens often have saprobic yeast phases, living freely, and these yeast phases can exhibit a decaryophase, unlike many other yeast groups.
Wow. So across the entire fungal kingdom, this yeast lifestyle just keeps popping up again and again.
Yeah.
So what does this all mean for us, wrapping our heads around it? It means yeast morphology isn't just fixed genetically, it's sometimes a direct response to the environment.
Absolutely.
Things like osmotic stress, high sugar or salt, or even levels of carbon dioxide and nutrients can trigger a switch between high-falling yeast forms. This incredible adaptability has evolved many, many times in different unrelated groups of fungi, making yeast a descriptive term for a form or lifestyle, rather than a single taxonomic branch. We're talking about convergent evolution on a microscopic scale.
That sums it up perfectly. And given this inherent complexity, modern yeast identification has become this sophisticated blend of traditional microscopy and cutting-edge molecular biology. Experts now differentiate yeasts by checking a whole suite of features: their minimum, optimum, and maximum temperatures for growth and for sporulation if they do it. Also, their ability to grow in various toxic compounds, their osmotolerance, which is their ability to grow in high sugar or salt concentrations. That's crucial for things like food preservation or fermentation industries.
Makes sense.
And of course, their detailed cell morphology and the specific method of conidiogenesis we discussed earlier. And critically, their DNA sequences, which offer the most definitive classification these days. There are even some quicker diagnostic tools, like you can differentiate many Basidiomycetes yeasts from Saccharomyces by a simple staining reaction with buffered diazonium blue B. Turns a different color.
Huh, interesting. It's just incredible how these microscopic, incredibly diverse organisms shape so much of our world, often without us even realizing the complex biology behind them. Yeasts have always been profoundly important to us, haven't they? Primarily as the producers of bread, a staple food across cultures, and alcohol, which, despite its dangers, remains our most widely used and accepted social drug. They're vital in countless food processing applications, too, from cheese ripening to chocolate fermentation, things we might not even think about. But as we've discussed, we also have to recognize their dangers, both in food spoilage, where they can cause significant economic loss, and critically, in the field of medical mycology, where some species pose serious health threats. Their impact on human affairs is truly profound and multifaceted.
Absolutely. And if we connect this all back to the bigger picture, I think the most important insight from Kendrick's chapter is that yeast is not a single unified group of fungi. Instead, it's a polyphyletic term describing a highly adaptable, often unicellular or dimorphic lifestyle that has evolved multiple times across the vast and diverse fungal kingdom. Their ability to switch forms, their diverse reproductive strategies, and their varied metabolic capabilities are really key to their ecological success and their enduring impact on our world, both beneficial and sometimes detrimental.
So, what does this all mean for you listening in? Well, next time you enjoy a slice of bread or maybe a glass of wine, or even, unfortunately, if you encounter a fungal infection, just remember that you're interacting with an organism that defies simple categorization. It's part of a group defined more by lifestyle than by strict ancestry. It really makes you wonder, doesn't it? What other common terms in biology might be hiding such a rich and surprising polyphyletic diversity, just waiting for us to take a deep dive? Thank you for joining us on this deep dive into the fascinating and surprisingly complex world of yeasts.